Introduce our presenter for today. Today we have Dr. Melanie Quintana, who is Vice President and Senior Statistical Scientist at Berry Consultants. Dr. Quintana is an expert in innovative clinical trial design, including adaptive and platform trials in rare and progressive diseases, and she has extensive experience working on clinical trials specifically in the fields of neurology and neuroscience, including the Healy ALS platform trial. So we are so grateful to have her with us today to present her talk, which is titled Clinical Trial Outcomes from Endpoint Selection to Estimand Analysis. Thank you, Dr. Quintana, and it looks great. You can go ahead and start when you're ready.
All right, thank you. And thank you to the organizers for inviting me to speak on this webinar. As was mentioned, I'm going to talk about clinical trial outcomes, but that involves often more than just endpoint selection. So also digging into what we mean by estimands and why careful thought around also the estimand and the analysis is important. I thought I would set the stage just by making sure we're all on the same page, defining what we mean by an outcome in terms of within a clinical trial. A clinical trial outcomes are really any variable that we can measure, and they're used to evaluate, of course, the effect of an intervention on health, safety, or experience. And important qualities of outcomes in a clinical trial are that A, they're predefined. They are something that we can measure and we can reproduce that measure, and that they are clinically meaningful in some way. Why do we care so much about outcomes, and why is thoughtful outcome selection so important? The outcomes that we choose really define success in the different phases of our trial. What does success look like in our trial? And once we select our outcomes, that can really drive the design, the sample size, and the duration of our trial. So it's really important to get that right from the start. Also, the outcomes that we select enable us to have regulatory approval or more or less clinical adoption based on which outcomes we select. And they can also ensure that we can achieve relevant conclusions for patients and all stakeholders. Now, the outcomes that we're talking about can vary across the development stages, and different outcomes dominate in different stages. But of course, within a different stage, you might include all of these outcomes. It's really which outcome is the focus of that stage. So in early stage development, we might be talking about safety, adverse events, lab abnormalities, things like that. In more of like a phase 2 stage, we might be thinking more about biomarkers or surrogate endpoints. Here we might be thinking about, MRI lesion burden, amyloid PET, NFL, things like that aren't the clinical efficacy outcome, but they're a biomarker or a surrogate for that outcome. When we move into stage 3, we're really thinking about clinical efficacy. So this might be functional rating scale, seizure rate, survival. We're really focused on how a patient feels, functions, or survives when we're talking about kind of clinical efficacy. And then in even later stages, when we're thinking about maybe stage 4 designs, we might think more about real world economics, patient reported outcomes, things like that. So this is really, which outcomes kind of dominate each stage. But as I said, many of these outcomes are included in every stage. For the purpose of this talk, I'm really going to focus on clinical efficacy outcomes and how those are selected. So that's really where the focus will be. Now, as I mentioned, this is more than just selecting the right endpoint. There are three, as I see it, three kind of important layers to being able to answer the question of interest or being able to address the objective of the trial. The first, as we said, is the outcome itself. This is what is being measured, the variables that's recorded. So an example here might be muscle strength. The outcome is muscle strength. But as we go deeper, the next level is something that we call the Estimand. This is not the outcome that's being measured, but what is the treatment effect that is being estimated? This is going to define the clinical question the trial will answer. It often also clarifies the population and how we handle what we call intercurrent events, events that might happen that might change that outcome or censor the outcome, like death in a trial. So an example of an estimand might be we are estimating our outcome is muscle strength, but what we are estimating is the treatment difference in the change from baseline, say at week 24. And then it might say something about how we handle intercurrent events. So regardless of treatment discontinuation. And then the final layer of this is how we get that estimand. How do we estimate the estimand? And that's really the analysis, right, that's tied around the outcome. And so this might, for example, be we're using a repeated measures model to estimate this. So just wanted to really highlight that there are multiple layers here and they're all intertwined. It's really the outcome, which is essentially change in muscle strength, the estimand, how we're estimating that treatment effect, or what treatment effect is of interest in being estimated, and then the analysis of how we're actually estimating that. I wanted to really kind of go through this talk by selecting 3 case studies, which I really feel kind of highlight these 3 different layers and how questions can arise in each one. The first case study that I want to look at is in a rare disease called GNE myopathy. Here, the strength in when we were designing this trial was that we had a really robust natural history data set. It had dozens of potential outcome measures. But the challenge was that there was really no precedent on what the best outcome was. So the challenge was really within that first layer of we don't even know what the best outcome is. We don't even know what our primary outcome should be. And then additionally, within rare diseases, as is often the case, we also need to think of and be a little bit more creative with how we're analyzing that primary outcome that we choose, because often clinical trials are really infeasible given the small sample sizes. So the challenge there in GNE myopathy that I'll kind of highlight is outcome selection, but also how can we develop more efficient analysis methods to make trials feasible within these rare settings. Contrasting to that, I'll talk also about case studies and designing trials in ALS and stroke. In both of these, the outcomes that you tend to use are very well established. So one might say, oh, this will be easy. My design is going to be straightforward. Everyone knows what outcome I should use in this case. And that's a strength. But just to highlight that there still are challenges that we have to think through. So for instance, in ALS, there's a big question on how we incorporate or handle the intercurrent event of death. Or do we even count it as an intercurrent event, or should it actually be part of the primary outcome? So we'll kind of dig into that challenge to highlight, there's more to this than just selecting the right outcome. And then finally in stroke, standard outcome is MRS. Huge question and challenge in how we actually summarize that outcome. How do we summarize that ordinal endpoint? And what's the right estimand for that? So starting with GNE, I will argue this is probably the most complex we'll start with. So I'll probably spend the most amount of time here just highlighting this. As I mentioned, the challenge here is there was no real consensus on what the primary outcome would be when we started this. And so the key question was really finding that endpoint that best captured progression in this disease and finding a really efficient analysis method that can make most efficient use of the limited data that we have. So the key takeaway here will really be endpoint choice and analytical efficiency are really central design challenges in very rare diseases like GNE myopathy. So what is GNE myopathy if you're not familiar? This is a rare genetic muscle disease. It's categorized by slowly progressive muscle weakness and atrophy. It affects different muscle groups at different stages of the disease. It often starts with the patient tripping more because their ankle muscles, their dorsiflex, start to become weaker. That can slowly kind of move its way up the legs and affect at first the lower extremities. And these MRIs, what you're seeing is a participant in the early stages of the disease might still have a lot of muscle in their lower extremities. But as they progress to later stages of the disease, you might really start to see almost all of that muscle turn to fat and atrophy. So the trial objective here was really, we wanted to discern in a clinical trial if the treatment slowed muscle strength, slowed the decline of muscle strength, because that is how this disease is categorized, right? It's categorized by muscle weakness. So did the treatment slow the decline of muscle strength was really the main trial objective. Now, the challenge in GNE myopathy is this is a very rare disease with really small sample size. There is very limited precedent. Progression is quite slow. So we're talking about 40 years going from initial progression to almost full progression. And the outcomes with the greatest precision to measure disease progression will really depend on the disease stage. So someone earlier on might be actively progressing in their lower extremities, but might still be quite ambulatory. Later on in the disease, they might be non-ambulatory and be progressing more on their upper extremities. So we're thinking more about like grip strength and elbow flexion and being able to use kind of those upper extremities. So the key question that we started with was really what outcome is the most precise in measuring disease progression across the broad spectrum, and which one is also clinically meaningful that could be the primary outcome in our study. As I mentioned, the strength of working in GNE myopathy was that we were very fortunate to work with a group of investigators at the NIH who had been collecting this wonderful natural history data set on participants, flying them into the NIH and collecting longitudinal outcome measures on numerous participants. At the time of the snapshot of this summary of this data set, they had about 38 participants. Following them every 3 to 6 months. We had an average follow-up of about 14 months, but some participants out to 38 months. And many different measures were being observed, 6-minute walk test, and then the adult myopathy assessment tool, several other functional rating scales, and then also what is called QMA. This is a quantitative muscle assessment for multiple muscle groups. Digging in a little bit more on QMA, because this will become central to the story, is this is, as I said, a quantitative muscle assessment. Here you can see a participant on the machine, and basically what the machine does is it records how many kilograms of strength are exuded when the participant uses that muscle group. Fortunately, we also have normative values, so there is a publication available to take a participant's age, gender, and BMI and get what we would expect, a healthy individual with the same age, gender, and BMI would score on this muscle group. So we could take all of those recordings and convert them to percent predicted, where, if a participant had 100% on a muscle group, that means that they're functioning at a sort of a normative level for their age, gender, and BMI. And then if they were, for instance, here where you look on left and right dorsiflex, they're more at about 25%. So they've progressed quite a bit on their dorsiflex compared to somebody who is of their same age, gender, and BMI without GNU myopathy. So this is an example of QMA. You can see that all the measurements are bilateral, and we can get them across a large range of muscle groups. So when we started this project, the first thing we did was take the natural history data and start summarizing individual measures. So this is an example of 6-meter walk test in meters. So you can see here that some participants, let me get the laser pointer. Some participants are scoring, very high and kind of stay there. So lots of variability and kind of baseline level of functioning, but not a lot of progression over the course of even 3 years. People kind of are where they are and they stay relatively flat on that over the course of 3 years. Then what we can do is take this data and produce summaries so that we can then determine, power calculations under this endpoint. In progressive disease, how we do that is we kind of take the data, we summarize, what is the mean rate of natural progression? That's the red line here. And then, if we were to have a treatment that kind of slowed that progression by 50%, that's what this green line would look like. And that's what we tend to power at. 50% is quite a large slowing. Actually, a lot of times in progressive diseases, we tend to try to aim for even like a 30% slowing. But here, these power calculations looked at a 50% slowing. And what we saw for 6-minute walk test is that this would take an infeasible number of participants. If we did a 1-year study, this would be over 1,000 participants. If we looked at a 2-year study, we're talking about 300 participants. To put this in context, we were really thinking about a trial with 50 participants that would be feasible to actually accomplish in a reasonable number of time. So both of these numbers kind of really not feasible for our study design. Another outcome that we used, and in a second it will come to light why I'm showing this exact outcome, is, we could take these QMAs and we can create composites with them. This composite was all of the upper extremity strength. So we're taking all of those kilogram units, adding them together for all the upper extremity strength. And again, kind of like 6-meter walk test, large heterogeneity in baseline values for this, and not a lot of progression within a participant over even 3 years. So again, because of this heterogeneity and slow progressive nature of the disease, again, a trial using this endpoint could be quite infeasible. Better than 6-minute walk test, but still needing about 500 participants for a 1-year trial or 120 participants in a 2-year trial. So another thing you can take from this is, if we're to follow people just for another year, you can really reduce your sample size a lot. So that is a whole nother talk and discussion of how you choose the right follow-up time. But just to note, that's also important here, that we noted, a 2-year study would get us a lot more power than, say, a 1-year study because of the very slowly progressive nature of the disease. So why did I circle and highlight this 1-year study with this specific endpoint? While we were designing this trial, Ultragenyx actually announced their top-line results from their phase 3 study, and they used that exact endpoint. It was in a study of about 89 adults. They were following them for 48 weeks, and it was not statistically significant. The study was a failure. But I highlight this to say it's really important to do this homework, because and to think about it in context of a reasonable kind of percent slowing that one might see in the disease. Because doing these power calculations, and we were able to do them because we did have the natural history data. Others might not have that data or that ability. You're able to see that using this endpoint, you would need a trial that was 500 participants. So The unfortunate thing here is, the question can't be answered if this study was just extremely underpowered for the endpoint that was chosen, or if the treatment really didn't work. So we look at the power across, many different potential endpoints. In summary, what we saw is that no single endpoint really provided us the sensitivity that we needed to run a feasible clinical trial. And the outcome selection is really challenging because of this heterogeneous progression and this slow progression, and the fact that it's going to occur across different muscles at different times in the disease. What we did take from all of this, and also meeting with the FDA and discussing different outcomes, is that QMA was determined to be the most direct measure of disease progression. And it also tended to be the most sensitive measure. However, even in using, different composites of QMA, we still weren't left with a single outcome that could be feasible in our trial. So in GNE, the central question was not just what to measure, although this homework kind of allowed us to make a choice on what to measure and what could be the primary outcome, but also how to measure it. So it really kind of shifted from what outcome should we use to, okay, we have to get more creative here with an analysis method. I don't want to spend too much time on this, but I do want to just give you a sense of creativity in innovative analysis methods and what you can do in very rare diseases. We don't have to just be stuck here and say, oh, well, my trial is just going to have to be underpowered. We can develop more efficient, innovative analysis methods that can help to allow for better power, especially in these rare diseases. In this example, what we did was we built what's called a disease progression model. A disease progression model is just a mathematical model that looks at how the disease progresses over time. It quantitatively captures that across single or multiple disease-specific biomarkers or outcome measures. In progressive disease, this is likely monotonic, meaning that we see this kind of decline over time and people don't decline and then jump back up. And this is just kind of an illustration of what this might look like in GNE myopathy for different QMA muscle measurements, where people might start to progress on which outcome over the disease span. And ideally, these are measured as a function of disease stage. This disease stage, or what we call disease age here, is likely unobserved and a latent variable, meaning that we need to estimate this. Where you are in the course of your disease is not something that we can get at a baseline visit. It's something that needs to be estimated from the model. So the idea here is that we take all of these participants that we have, that are kind of short snapshots of what the disease course looks like. And we can think of these as kind of small puzzle pieces of the disease. And we can put them where we think they fit in the puzzle based on this kind of latent disease stage parameter. So this is an example of that, just kind of taking 4 of those snapshots and putting them together in the puzzle. If we do this with all of the data that we have, now instead of only knowing something about 2 or 3 years of the disease, when we fit everyone together, we can get that entire characterization of the disease across, say, a 30-year span. So we put all of this data together and really try to understand how participants will progress on each of the endpoints over the course of the disease. So we were able to kind of fit this to the natural history data and get these summaries. What was really cool is before we went into this, we were just doing the modeling and we showed this plot to the investigators and they said, oh, this makes complete sense. Prior to this disease was called the quadriceps bearing disorder, meaning that knee extension we don't see much of a progression on that outcome. So that's why that gray line was relatively flat. So why am I telling you about this natural history disease progression model? You might be saying, I thought we were going to talk about how you should analyze clinical trial and determine if a treatment actually works. Well, you can apply this same concept to clinical trial data. And I'll just give you a little illustration of why that might be really beneficial. Here, we can use this type of method to really get a more powerful analysis tool. Here in this illustration, this is one muscle group. We have muscle strength plotted, simulated muscle strength plotted for, think of the gray data as placebo data, and the blue data is a treated participant. You look at the data in this way. This is looking at their muscle strength over length of follow-up on over 2 years. And it's really hard to tell, is this blue participant progressing slower than we would expect? They're in the middle here of a bunch of noise, and it's hard to distinguish that noise and determine if there's some slowing in progression. But if you use the same concept of aligning these individuals, these short snapshots in the course of their disease, you can really explain a lot of this variability. So we go from a picture that looks like this on the left to one that looks like this on the right, where now we're explaining a lot of the variability because of disease stage. And we can see a much cleaner picture of progression. And what that allows us to do is then compare that treated participant to similar disease stage participants in the placebo group. And now you can start to see, maybe there is some differentiation here. Maybe that treated participant is demonstrating a slower progression than what would have been predicted for the same disease stage in placebo group. So this is just a quick illustration to kind of demonstrate why these methods can be more powerful. Doing something as simple as adjusting for disease stage can really kind of reduce your variability and allow you to have greater precision in estimating your treatment effect. Additionally, this is another example of a participant. This is an earlier stage participant. And if we were just to look at their values on knee flexion, in that on that endpoint, we are not expecting much progression because we think they're at this stage in their disease where that muscle is still performing quite normal. And actually, when it's performing normal, it tends to have a little bit more variability. So we don't expect to see much differentiation on knee flexion at the earlier disease stages. But what endpoint we do expect to see a lot of differentiation on is dorsiflex. That was the ankle. That's the one that progresses earlier on. And so someone who's earlier on in the disease, it might make more sense to look at their ankle strength to see if there's any differentiation between the treated groups and the placebo. So it takes a lot of work to kind of build these more innovative analysis methods that make sense for the disease that you're considering, but it's entirely worth it. So we went from a trial that had about 10 to 40% power, depending on the single endpoint you chose, to having over 80% power using this more innovative analysis method. And this trial did run through NeuroNext, and this analysis was used and entirely appropriate to get that additional power. So just coming full circle with GNE myopathy, what does this look like? What did this look like for this actual trial? Well, the outcome was actually, I should have put in parentheses here, an S, multiple outcomes, were the bilateral muscle strength and ankle dorsiflexion, knee flexion, knee extension, shoulder abduction, elbow flexion, and hand grip. So it was actually multiple outcomes, and it was being measured by QMA. The Estimand was the change in the rate of disease progression, common change in the rate of disease progression across all muscle groups in the treated participants relative to the control. And the analysis method that we used to estimate that was this Bayesian disease progression model. So now let's move on a little bit to the ALS example. So contrast the GNE myopathy example where our challenge was really first selecting an outcome and then building kind of an innovative analysis method to an ALS example. Here, when I started working in ALS, I thought, this will be easy. They already know the outcome that should be used. But then when you really kind of dig in, you realize there are other challenges here. And the main challenge in ALS, because it is a fatal disease, is how to incorporate or account for censoring due to death, and how to choose an aligned analysis to that Estimand. So the takeaway here is, even when you have really established endpoints, there's still a lot of challenges that can remain in defining that Estimand and the optimal analysis strategy. So as I said, this was well established. FDA actually has a guidance in ALS, and they recommend as the primary outcome using something like this ALS-FRSR. This is a questionnaire, and it incorporates 12 different items ranging from speech to respiratory insufficiency, things like walking, climbing stairs. Each item a participant can score anywhere from 4, which is normal abilities, to 0, which is completely, no abilities within that item. So overall, we tend to take all these items and total them. And so a score of 48 would be completely normal in all of the items, versus a score of 0 when you've completely progressed and you have no ability on any of these items. The FDA guidance, and where the complexity comes in, went on to state, if patient function using ALS-FRSR is intended to be assessed by the primary outcome, mortality should be integrated into the primary outcome by an analysis method that combines survival and function into a single overall measure, such as a joint rank test. So the question now is, okay, is the outcome ALS-FRSR, or is it ALS-FRSR and survival? In terms of the question for the Estimand, if the outcome is just ALS-FRSR, how do we handle death as an intercurrent event? Or if it's the combined ALS-FRSR and survival, is it part of the Estimand, and how do we incorporate it? And then for the analysis method, which method do we use that kind of aligns with that Estimand? Is it a functional only model if we're just thinking of ALS-FRSR as our outcome, or, as the FDA recommends, something like a joint rank test, or even a joint model like we just showed you in GNE myopathy? So really the principle here is that you would choose the method that kind of went along with the Estimand that was chosen. To answer these questions, you really have to start with the clinical question of interest. And so the two options really are we going to go with an approach that is function focused, or will we go with an approach that incorporates both function and survival? If we're looking at function focused, we're interested in assessing the treatment effect only on function. And then so our example Estimand might be change in ALS-FRSR in the treated compared to the control group, but it still needs to appropriately account for death, and we'll get to that in a second. Or option B could be, we actually want, multiple outcomes, function and survival. And then an example Estimand might be, differences in rates of progression of function and survival in the treated compared to the control. But we still have a lot of questions here. Is the trial successful if it shows benefit on either endpoint, or does it need to show benefit on both? And, what if we show a strong benefit on function, but no benefit on survival? How should those trade-offs be taken into account? So those are the questions that we kind of had if we had multiple outcomes here. First, I want to dig in just for a second on why we care so much about this, and why, if I want function as my only outcome, why do I care about this Estimand question, or how we handle death? Well, functional progression is measured repeatedly over follow-up. deaths, which happen and occur in clinical trials in ALS quite commonly, create missing data, but it's not random missing data. It's informative missing data, because what tends to happen is that faster progressors are more likely to die over the course of the trial. So we need to account for that in our how we estimate things in our analysis method and our Estimand. And this is just a short illustration of this. Here's a plot that I created through simulation. The blue dots are the treated group. The red dots are the control. Remember that high value is good here for this outcome. So you see the blue dots are higher than the red dots at the end of a study. So if you saw this plot in a manuscript, I agree we should have some error bars here on these dots. Would you think that the treatment slows progression? I think all of us would. We would get excited about this. But what the truth of this simulation was is that they actually had the exact same progression rate. But what I did in simulation was just had the treatment group have more deaths. So there must have been a safety issue here with this treatment. It actually led to more deaths, and those deaths occurred in the faster progressors. And so it looked like there was a slowing in the progression rate. So this is just a simple illustration to show, why we make such a fuss over this. Why do we care about this so much, even if we're just analyzing ALS FRSR? So hopefully I've at least convinced you that Estimand's an analysis strategy. Even if you're doing a functional only analysis, you need to think carefully about those things. Additionally, as the FDA recommends, you might also think about choosing, the combined question. We want a method that integrates function and survival. We want to look at the combined effect on both function and survival. And within ALS, there are several different strategies that appear in literature. One is this joint rank method, which actually is a hierarchical composite of these two endpoints. You may have heard of a win ratio or CAS endpoint. And basically what it does is it ranks each participant to every other participant, and it ranks them based on, first, their time to death, if they've experienced death over the course of the trial. And if they did not, it ranks on the functional progression. The challenge with this is that it completely ignores functional progression if that participant died during the course of the trial. So you might have a participant who died one day earlier than another, and they would rank less than that participant, but they lived a life and their functional status remained well above that patient during the entire time they were alive. And so that's really where the challenge comes in with the joint rank method is that appropriate, especially in ALS? So another approach that is gaining popularity is joint models. And this is very similar to what I just went through with the GNE approach, that you actually model both function and survival. The challenge here is, how do you combine these two endpoints into a single test of, does your treatment work? And so there are different approaches for this. One might, for example, sort of estimate a common slowing in progression across the two endpoints and test that to see if that leads to a beneficial treatment or not. So the question you should be asking yourself is, how do you choose? Other than starting with the clinical question of interest, how we choose is very similar to what we did in GNE simulation. Simulation can really help us choose what is the best approach in our study. And you want to make sure you take into consideration a lot of different things. What is the clinical consideration? What's the study length? That might impact the mortality rate. What's the study population? What is that correlation between function and survival? And where do we expect the treatment effect to be? So for example, if you're running a 24-week study where a mortality rate is expected to be very low, is this fancy joint model even necessary or appropriate? And these are questions that you can answer through simulation and really kind of considering this quite carefully. Okay, just to bring this all back around in ALS, One example of what these strategies or these layers might look like is, potentially, and this is actually the example from the Healy ALS platform trial. The outcome there is, we say, change in disease progression from baseline through 36 weeks as measured by ALS-FRS-R survival. The Estimand was a slowing in disease progression common across those endpoints. And then our analysis was the shared parameter joint model. And I've provided references at the end if you want to dig into this a little bit more. Finally, I just want to take 2-3 minutes, and I won't take much time here, to talk about another area which is kind of similar to ALS, and that's stroke. Here again, we have an established functional outcome, but the challenge is really, again, how do we analyze it? How do we summarize it? There's a lot of questions being asked. So common options are, we see a lot of studies where they're dichotomizing this ordinal outcome. Another option is an ordinal shift analysis, or you might apply weights to the different levels and do a weighted analysis. So it's really important, even though the outcome itself is well established, to carefully think through what is the Estimand question that you're interested in answering, what is the clinical interpretation if you say dichotomize versus don't, and then what analysis method should I use? Often that analysis method will follow your choice in summary. If you're dichotomizing, you might use a binary model, or you might use an ordinal model, or weighted analyses might be different depending on how you're summarizing the outcome. This is just one example from JAMA recently. This study did dichotomize, and it looked at the MRS score of 0-1. It saw an increase in 0-1 scores for the treatment compared to the placebo group. But I guess the question I want to leave you with is, what are we missing with dichotomization? Well, we're missing summarizing what's happening across the entire scale. And I don't want to spend too much time on this, but I'll just say a lot of my colleagues are quite passionate about this and have thought about this in a lot of detail. So if you're interested in this topic, there's 2 great podcasts that you should listen to, one by Scott Berry alone, and then one by Lindsay and Scott. And you can get a sense of where they're leaning based on some of these titles. But I urge you to kind of listen to that. And it's another great example of a disease area where you think, okay, design here is going to be easy. The outcome is known, it's going to be MRS. But there's a lot of design questions that still remain, and it's important to do careful consideration of those. So just in summary, to design smarter trials, I'll say one piece here is really that natural history studies are invaluable. They allow us to do kind of evidence-based simulation, and that can really allow us to understand the power of our designs under different endpoints and evaluate feasibility. And I think the GNE example really highlighted that of being able to establish there is not one outcome that we are going to be able to use to be powered for, and we could establish more innovative approaches. In rare disease, especially, being able to use the data as efficiently as possible is important. However, when we're doing more innovative things, you really need to discuss the novel outcomes or analyses with all stakeholders early on. This is incredibly important to get buy-in from everyone early on. And then the last thing I'll leave you with is that, partnering with experts is really important. Strong statistical collaboration is essential, not just saying, hey, I have this grant. Can you tell me what the outcome should be and the sample size should be? I need it in an hour. All of this takes a lot of work and a lot of thoughtful consideration. So really partner with those experts early on, and ideally partner with somebody who has an understanding of your disease context. I'm not saying if you're in ALS, you need an ALS statistician, because those might be harder to come by, but maybe find someone who's done something in neurodegenerative diseases who can really understand the context well. And then I will leave you with some references. We can also pass around these slides to anyone who's interested in digging in a little bit more on some of these topics. And I will stop there and open it up to kind of questions.
Thank you so much, Melanie. That was fantastic talk, and excited to get into some questions here. We have a couple already in the chat, and then folks are also welcome to raise their hand, but I'll start off with the first one in the chat, which is, was this work on disease age and natural history GNE published? Was the Ultragenyx 89 patient study positive when analyzed, taking into account disease age?
It was published, and that was part of the references. So we have published both the progression model in GNE myopathy, and within that publication, we talk about kind of how we might apply this to a clinical trial. And actually, we have now applied this to a clinical trial. It was actually just presented at AAN. And that publication on the actual implementation of that trial hopefully will be being put together soon. The question about Ultragenyx is, unfortunately, we didn't have access to that patient level data. I would love to get access to it and actually apply the disease progression model and see, if there were any new findings there. Great question.
Thank you. All right, there's another one in the chat. Can you clarify the definition of endpoint in relation to Estimand and outcome?
Oh, yeah, I think I kind of used endpoint and outcome interchangeably. But it's certainly, I used it kind of interchangeably with outcome. The endpoint, I would say, is, yeah, the thing that you are measuring, while the estimand itself is more about the treatment effect that you're estimating. That helps.
Thank you. Other questions, you can feel free to unmute and ask, put in the chat. Feel free to bring in a topic specific question if there's a trial you're working on that you have a question about.
Yes, please.
All right, here's one. In these rare diseases, when starting a trial, often the natural history isn't available. Do you recommend pooling natural history with the treatment arm, even if done while the trial is running?
Okay, let me think if I can think of what is meant here. I think you could pool the natural history data with the control arm, potentially. I don't know if that was the question, meaning doing actual comparisons to natural history data and using that as maybe an external control. Are you, if we're thinking about kind of pulling the trial data with the natural history data to kind of guide decisions in the trial while the trial is running, I think that would be a little bit harder because you might then have to make trial adjustments. And that's kind of harder to do in a pre-specified, when we would like to have all of those decisions kind of pre-specified from the start. Yeah, I agree that if you don't have that natural history data from the start, it can be quite hard to try to answer some of these questions. Ideally, then potentially you have some publications where summaries were provided, and you can use some of those summary statistics from publications to kind of make these evidence-based decisions. But yeah, I have to say, I think the first step in any kind of rare disease with a lot of unknowns should be a really nice natural history study. And if you can collect the data in your natural history study in the same way that you might do it in a clinical trial, that would make it, even better. And then potentially you're able to kind of use that, especially in a rare disease, to supplement your control data.
Thank you. That's great. And helpful comment in the chat. I bet FDA guidance may be a helpful starting point to think about how industry has compensated for the rare disease scenarios.
Yeah, that's right. And FDA just came out with a new draft guidance in Bayesian statistics, and they talk a lot in there about, especially in rare diseases, how we can utilize external data. And so that's a great place to start. If you haven't read through that, I highly recommend it.
Okay, we've got another chat question. So with a sample size reduced, Are any of these rare disease trials actually truly powered when they start, or do they just turn out to be underpowered but have statistically significant outcomes?
I think it depends. I mean, that depends on the disease, the outcome itself. Um, I think, and the treatment effect, right? So if you can see really large treatment effects, you can be powered with quite small sample sizes. So that just really depends on the specific disease.
Great. What other questions do people have? Any follow-ups to any of these? We have a couple more minutes. If anyone has questions. All right. Well, thank you so much. It was such a great talk. We would love to share the slides if you're willing. I can post them with the recording. And the recording will be made available. One in the chat, it says, could you offer the punchline for the 2 talks you gave QR for?
Yeah, the punchline is along the lines of the JAMA article I showed of um, uh, dichotomization not being the best, not being a great idea in terms of these ordinal endpoints. Dichotomization is easy, right? And we think we understand it. It's easy to look at the proportion of zeros to ones versus a worse outcome. But you're removing a lot of the information when you're doing that. You're missing a lot of the information. So there are better approaches. One of those could be, weighting the different outcomes. And they talk a lot about these weights that can be applied to the MRS, and they can be clinically meaningful weights. But a lot of times, there could be pushback because, are those weights clinically important, or does everyone agree by those weights? And then the fallacy there is if you're dichotomizing, you're weighting, right? You're giving a weight of 1 to the zeros and ones and a weight of 0 to everybody else. So that's kind of the fallacy of a lot of people kind of have issues with these weighting approaches, but by dichotomizing, you're actually weighting and you're assigning a weight that might not be clinically meaningful. Kurt, I see you're on chat and maybe giving a better punchline than but
Actually, I typed it while you were saying the exact same thing. So I apologize. You got right to where I wanted to go. So
yep, yes, exactly. But I urge you to listen to it. Scott feels very passionate about these things, which a lot of us do, and it's a great podcast. So if you have the time, listen to it.
And here's another good question. What would you do where an outcome starts out as unmeasurable due to severity and then improves naturally? So, for example, visual outcomes following optic neuritis.
Yeah, that's a great question. And that kind of leads into how do you handle outcomes how do you almost like impute a value for an outcome that can't be measured? We had this issue with QMA, right? Like when someone can't do any force, is that a 0? Do we impute that as a 0? So yeah, I would urge you to kind of think about what value you would assign to those kind of unmeasurable outcomes. And it's really important to think about how you collect that data. Also in GNE, we had to kind of go back in and really dig into was it missing because they were unable to do it because of disease severity, which could get a different value assigned, versus missing because of another reason. So I urge you to really try to think about how you'll collect that data and then think about the value that you would assign at the beginning if it's truly unmeasurable due to disease severity. But there are ways to handle that.
Thank you.
Yes.
Any
And
last questions? Go ahead.
happy to provide the slides. We will send them over.
Thank you. Well, it was another great session. Thanks to Melanie. We have our next CTMC webinar will be in 2 weeks on May 8th, and that is titled What to Know About Sample Size Before Talking to Your Biostatistician. Always a great talk from Dr. Sharon Yates, so we look forward to seeing you all again soon. Have a great
Thanks, guys.
day. Thank you, Dr. Quintana.